30 seconds usually.
I think we're live.
Okay, I think we are live. Well, welcome everyone. My name's Daniel. Tonight we've got Jocelyn Downey, and we're going to be discussing vaccines. Obviously, we usually have Phil here, but he's otherwise engaged this evening, so you just have me, I'm afraid. The reason we've tried to rush this one through to get this done because one, Jocelyn knows what he's talking about, and secondly, we've all got a lot of questions about vaccines and constantly getting a lot of questions from friends, family, and there's a lot of stuff online about vaccines, a lot of misinformation, disinformation, and all sorts. So I think it'd be really good for us to. I've got a lot of questions I want to ask Jocelyn. I've got a lot of questions that have been given to me as well, and also hoping that during the live stream we'll get some more questions as well, and we're happy to. Jocelyn's happy to try anything, well, as long as it's related to vaccines. And so I guess, Jocelyn, why don't you just tell us a little bit about your background and why should we trust anything you say about vaccines?
That's a brilliant question to start. So I did an undergraduate degree in science, and then I went on to do a PhD in genetics. And after which I embarked on a career in immunology. So I went to the US, worked at the Scripps Research Institute in California for a while, doing work on septic shock. Came back to the UK, worked on tuberculosis, worked on HIV, worked on what they call cell signaling, which is looking at how immune cells signal to each other, particularly looking at T cells. And had some involvement in HIV vaccine trial, which gave me some authority. I mean, I can't speak, obviously, I have knowledge, quite a wide knowledge of immunology, but there might be areas where my knowledge is weaker, which I think I put in my blog, All academics have a speciality, and you move away from the speciality, you suddenly find that your ability to speak comprehensively deteriorates. So, and the HIV vaccine trial is looking at not using a vaccine to prevent HIV, looking at a vaccine to help HIV patients to have a better immune system. So that was using a DNA vaccine. So I have some similarity with using DNA. In fact, it's not RNA vaccines, but they're a later development. Yeah. So, yeah.
Good. Okay. So, one thing I think a lot of people know about vaccines is that they've been, pretty successful. So we managed to eradicate smallpox, which is great. We're very close, I think, globally. Correct me if I'm wrong, to eradicating polio as well. I think that's the sort of next one that we're closest to getting rid of, which would be which will be great. So could you give us a sort of for everyone listening, sort of a brief sort of historical overview of vaccines? Really, I think would be quite helpful for people.
Yeah. So even before vaccines, we have a treatment called variolation, which was for smallpox. So we see incidences in the 1500s, maybe in China and in Asia, where people would take some of the crusts from the smallpox wounds and give it to somebody, hoping they would get a milder form of smallpox that would then treat them. And that was used, that was developed. We don't know when it was originally developed, but it was certainly introduced into the UK in about 1715. And then, but the problem with that was that it has a high risk of people developing more severe smallpox and dying from the disease. And then in around the 1770s, Edward Jenner noticed that women who had cowpox, sort of the disease, very mild disease, didn't get smallpox. So he did an experiment where he took the cowpox pustule, gave it to a young child who survived, then challenged with smallpox. And that was the beginning of the first vaccine. So the cowpox virus became then the first vaccine that was distributed worldwide. And then 1840, variolation was banned because of the high risk. And cowpox became very widespread and led eventually to the eradication of smallpox. So that's the background of smallpox. And then we had to wait a while for the following vaccines to develop, vaccines against rabies, measles vaccines. There have been attempts to do vaccines for quite a number of organisms since then, but some are more difficult than others. So that's the beginnings of vaccination. But that was sort of the early days, really, before immunology as a field really got going. So as later on, we started understanding more and more about how the immune system worked and how the body fought off disease.
Okay. So how do vaccines sort of normally work? And then what is, I think what a lot of people are asking is, especially people who would normally have a normal vaccine, say for something for TB or for MMR and things like that. Mm-hmm. So how does the new sort of Pfizer vaccines and Moderna actually differ? We hear about this mRNA vaccine. Yeah. So how does a normal vaccine work, and how do these ones differ?
Well, I guess the idea of a vaccine originally was you with like a mild form of the disease, either take a dead form of the disease, kill a virus or kill a bacterium, and then when you inject it into somebody, they would develop the same immune response that they would develop to the live organism, but you wouldn't get all the symptoms of the organism. So because the organisms are either weakened, because using a related organism that's close enough related that it would stimulate a similar immune response, or a killed version of the organism, so that you're hopefully generating a very similar immune response to how you would have immune response to the live organism. But since it's dead, you wouldn't get none of the symptoms of the disease would appear. So that's the basic principle behind them. The immune system itself has a lot of layers to it. I think many people consider the antibody response to be the response that they think of when they think of the immune system, but in fact there are different strands to the immune system. So, some of those are simple as the barrier, so your skin preventing infection organisms getting into your body, or the mucus that you have in your mouth and your lung lining that prevents viruses from sticking to the cells. So that's a sort of barrier to the infection. And then
okay, so go on. Go on, Karen. Sorry.
Then you have the antibody response. Then you have cellular responses where cells themselves attack infecting organisms. And then you have what's called innate immunity, where you have cells in your body have receptors that can recognize foreign organisms and mount an immediate attack to them. So you have a number of different layers to the immune system.
Okay. I think a lot of people are aware of DNA, but what is RNA? So when we talk about mRNA virus, could you explain RNA in a way that we could maybe help people understand it a little bit more clearly?
Yeah, I think so. Every cell in your body has DNA in it, the same DNA. You hear the chromosomes you have, and that's every cell in your body has basically the same chromosomes. However, every cell has to have a way of regulating which part of your DNA, which part of your chromosomes is switched on or off at any time, because you don't really want to have, as I used to say, eyes in your feet. You want to have the genes that are switched on in your feet have to be different from genes that are switched on in your face or any other part of your body. So the way the body does that is when it wants to turn a gene on, switch on, it makes a copy of that gene, not into DNA, but into a molecule that's closely related to DNA called RNA. And that molecule then leaves the nucleus of the cell, enters the cytoplasm of the cell, into the main mush of the cell, and where it is fed into a complex of machinery in the cell called the ribosome, which literally reads the code of the RNA molecule and says, oh, this RNA molecule is this protein, the manufactured protein that's coded by the RNA molecule. Yeah.
Okay, so I guess get on. That's a bit of the background. A lot of the criticism that I've heard about the vaccine. I should say at first that I'm very pro-vaccine. Just so I'm just presenting some of the questions. So a lot of people have argued that, and this is even from healthcare professionals as well. This is a lot. I think a lot of people have a concern that they feel that the vaccine has been rushed. And are worried about the kind of maybe not the efficacy in terms of whether it works or not, but in terms of its safety, that actually it's gone through phase 1, phase 2, and phase 3 trials quite swiftly compared to any other vaccine. And that's the case. Yeah, it's vastly quicker. I mean, so I guess one is, should we be worried? And two, how much quicker has this been than the average vaccine that comes onto the market?
Oh, there's no question this vaccine has been developed very quickly. I think it's much quicker than any previous vaccine. There are different aspects to why that has happened and why you may or may not be concerned about that. The technology that's used in the RNA vaccines, or even the Oxford vaccine, which is a virus vaccine, has been in development for some time. The idea of using viruses or DNA vaccines goes back to the 90s or earlier, but I remember the 90s. And use of RNA as a vehicle for vaccines is really developed in the last 10 years. So there's been a sort of phase of development of how we can assess the vaccine safety. Now, injecting someone with a live organism, so if you were to take, say, COVID and kill it, which has been done in one trial, and inject it into individuals, there are a number of risks that go with that. One is you have to guarantee you've killed the organism. You have to guarantee that the agent you've used to kill the organism doesn't kill anybody or cause any potential harm to a person. And that alone can take some time. So the advantage of an RNA vaccine or a DNA vaccine is that you can take only a small part of the organism. You don't have to use the whole organism. You can use the smallest part of an organism that you know is important to that organism's function or its immune response. In this case, they've used the spike protein of the coronavirus, so which is involved in binding to cells. So the idea is that they can block binding to cells, or they can mount an immune response to what is very obvious target on the virus. The technology in terms of specifically in terms of coronavirus, we had the 2002-2003 SARS outbreak, which caused a bit of a shock to a number of people. And it was contained within really Far East, for the most part. I think 9,000 cases altogether, but it was largely contained in Guangdong province in China and then Hong Kong. But that and then the MERS outbreak in 2012, both of which are human coronaviruses, made the academic world and the pharma world very concerned about the possibility of a coronavirus pandemic in the future. Because while people talk about there being a lot of coronaviruses, in fact, there are only 7 human coronaviruses. And 3 of those appeared in the last 20 years, SARS, MERS, and now COVID. So there's been a massive concern of the potential of a quite serious pandemic worldwide so for that reason, a number of companies such as Moderna were already preparing technology that would allow a candidate vaccine to be produced quite quickly for a coronavirus. And in this case, so the spike protein is something that's conserved. The spike protein, or variations of it, you see on MERS, SARS, and COVID. And it's an obvious target for a vaccine. So really, in January, as soon as the sequence was published, the virus genome sequence, the RNA sequence, was published in journals, people were immediately saying, okay, let's take the spike protein and investigate that as a possibility for a vaccine. In fact, I think at the end of January, there's even BBC reported one company was already doing that. So there was a very quick response to develop a vaccine. Now, because it's only part of the virus and not the whole virus, there's no chance of infection. And it immediately reduces some of the risk factors that are associated with developing a vaccine. There was also, and it is true that the process happened very quickly. And it's important that we are very careful. There are a number of things we have to be very careful of. We have to see, is it safe in humans? Does it reduce the symptoms of disease? Does it prevent it spread from one person to another person, which is really a separate issue? And are there any long-term effects? Does it produce long-term immunity? So these are all questions that have had to be answered quite quickly in an effort to try and find a way to stem this pandemic. Now, some viruses that take a much longer time to develop a vaccine against, but because of the nature of the virus, some viruses mutate very quickly. We're very fortunate that while this coronavirus does mutate, it seems to be a much slower rate than some other viruses. Slower, for example, than influenza, and much slower than HIV, which mutates very quickly. So HIV vaccine trials have been hampered in part because of its fast mutation, and by the fact that the HIV virus can actually integrate into your own DNA, which makes it another challenge, whereas this virus does not. So there are, that's been helpful. There have been scaling up of various processes in the last 20 years, looking at our knowledge of proteins structures, which has been greatly improved. And I think also this technology has just been helpful in terms of quickly finding a solution, the answer.
Okay. So how rare or common are vaccine, side effects to vaccines, and have there been any instances of individuals who have developed, serious side effects months or years later? Anything documented? I mean, my thinking is, there's no precedent in the sense that even if that is the case, which I'm sure you'll explain in a minute, that's with the more conventional live viruses compared with this is the first one that's been used, an mRNA virus. We don't have any precedent for it. So,
that's a big question. So every disease is different, and because nature each viral infection in fact is different, that can create or reduce risks for any side effects from a vaccine in a sense. So with some diseases, there might be a concern that giving a vaccine might increase risk, of developing serious symptoms from the disease. So there have been concerns in some vaccine trials in the past that, such as dengue fever was one classic example, that perhaps giving a vaccine to dengue fever because of the nature of the virus, rather than prevent the disease, might actually make first-round disease worse because of some of the immunological targets of the virus. So that is one concern. BCG is a vaccine that's been studied very thoroughly. BCG is a vaccine against tuberculosis and, to a lesser extent, leprosy. And it was studied very widely in the 50s and 60s as a vaccine candidate. It's proven to be very safe. But that doesn't mean that it works everywhere in the world. So when the studies were done in the 60s, it was discovered that in some parts of Southern Africa and in 2 states in the US, the vaccine didn't really have, it wasn't really effective. And there were concerns that in Southern Africa, having the vaccine might actually make it more likely to develop tuberculosis. But that wasn't necessarily because of the vaccine itself, but because of other organisms in the environment that were altering the effects of the vaccine. So that's an ongoing question, actually, with the BCG vaccine. In terms of long-term effects, there are some, I'm not aware, but that doesn't mean I don't, I'm trying to think of a vaccine trial where there's been long-term effects long time after the vaccine.
That would seem quite unlikely to me.
So unlikely. I can think of a couple of questions related to that. One of which would be effects on your mood, effects on, whether it could cause long-term. Some vaccines could potentially cause changes in mood or changes in chronic fatigue. I don't think there's any certain information about that, though, to my knowledge.
Okay. So again, I think a lot of the questions are going to sort of related around the Pfizer vaccine, because that's the one that's now been approved here in the UK. Another question I've heard asked is that if you look at how do the number of participants involved in the Pfizer phase 1, phase 2, 3 trials compare with with other vaccines that have obviously gone through a much longer and some would say rigorous process, which I think that's the concern that people have, is that which I think I'm you know, I don't think it's been less rigorous. I think there are really good reasons why the context of COVID globally, of a pandemic, has allowed us to develop a vaccine this quickly. But I would be interested to know, I mean, like, so say from other sort of vaccines, you know, how many would you normally hope to have in a trial to demonstrate efficacy?
Yeah, well, so efficacy versus the number of patients that I've seen enrolled on trials, up to 10,000 in each of the trials, is reasonable. Now, I have to do a comparison to in 2011, there was a vaccine trial that was announced worldwide as the world's first HIV vaccine trial. They're called the RV144 vaccine trial. And that was undertaken in Thailand. I think there were 16,000 individuals enrolled in that trial. So this vaccine is very comparable to that. It was, it's conducted internationally in different countries. And it's really looking at comparison, which you can't give someone a vaccine and then give them the virus to see if it works. That's not ethical. So we have to look at incidences of people who have had the vaccine who then go on to develop COVID in the vaccine group and the non-vaccine group. And the number of patients seems to be very comparable. Although obviously vaccines that have had a much longer, vaccines that like BCG that millions of people have had, that's almost an ongoing study. And this is still fairly early phases, stages of the vaccine. Yeah.
So your answer would be that actually it's comparable to other vaccines, I think. And that's what I said. I mean, that's the criticism of people, is they said, maybe it hasn't had enough people. But actually, if no one ever says, well, what is the number? What's the threshold that you would accept to demonstrate efficacy and safety of the vaccine? But I haven't seen it. It's something that fits on a meme, rather than that.
It's in the company's interests not to develop a vaccine that's going to cause long-term harm, to be honest.
Absolutely. Although this is why people say that, again, another claim is, why don't Pfizer have indemnity? That the government has given them an indemnity. But if you look at numerous countries have it's not just this, flu vaccines. A lot of countries give indemnity to pharmaceutical companies. Otherwise, they would never, given the risk involved, they would never end up. It wouldn't be cost effective to develop, vaccines.
Imagine it bankrupt them if they had to then pay insurance. Yeah.
So you wouldn't have anything to add on that in terms of?
No. I mean, I did see that the government given indemnity, and that's understandable given. But I don't think that's necessarily a reason to doubt the effectiveness of the vaccine. Yeah.
No, I think what's happening now is people are starting to. I think before COVID, a lot of people didn't know much about vaccines, and now we're learning things about the COVID vaccines that we think sound bad, even though there's precedent in the other vaccines that people have already had or would have if they needed. So in terms of the indemnity, indemnity that's always been in place in a lot of countries for a lot of vaccines. But I think hearing about it now, spun within the web of a sort of anti-vax narrative, kind of lends that. It's like, oh well, if they're having indemnity, that means that maybe they don't know it really works, and actually they've got nothing to lose if it doesn't work. But actually, Pfizer and Moderna have a lot to lose. I think Moderna especially was one. I think in the US chose of one of the 6 vaccines. I think that they funded the development of actually have a lot to lose, because as the smaller companies, this is their make or break. If they do well in this, well, this is going to make them for generations. Whereas if they mess up and get this one wrong, that's the end of the company. Realistically,
and that's government as well. Yeah, I mean, you get lots of trust in both in the companies and in government, which would do nobody any favors.
Yeah. Okay. So I had a question earlier from someone who'd asked about. They said that the Pfizer seemed like they were surprised by the anaphylactoid reactions to the vaccine. I think was it 2 or 3 individuals who had some?
I think reactions. It was something else emerged that I don't know about.
Yeah.
That's true. Yeah.
So is that my understanding is that people who are already my understanding is these individuals have already had allergies and to other things, and usually it's pretty universally known that if you have allergies, you're much more likely to have allergic reactions to other sources. So is this should we be surprised by this, or is this absolutely normal?
It was very sad to see that 2 people had developed a reaction against the virus, the vaccine. Sorry, it's relatively unusual thing to happen. Now, the vaccine itself is very simple. The vaccine consists of RNA, which is a molecule that's natural in your body, and then it has with it sort of a fatty lipid molecule that helps to carry that RNA into your cells. And it's possible that the people that got that vaccine and reacted to it reacted against some components in that lipid carrier, for example. I think one of the components of that was polyethylene glycol. And polyethylene glycol is used very widely in food, so it's fairly safe. And we use it in labs to preserve cells. It's quite safe, but occasionally someone can react to it. Very, very rarely, but it's possible. So for people that have an allergy to potential components of the vaccine, that's very sad, but I would imagine it's a relatively rare event. I wouldn't expect that to be a common side effect.
Okay, so it shouldn't be something that should be a worry to people. I mean, I know that they've recommended that people with certain allergies should not have the vaccine.
Yeah, I think if you've got a history of allergies or serious allergies, to which the point where you're carrying an EpiPen, maybe you have to consider talking to your GP about whether or not you take the vaccine. But for the majority of people, the vaccine really shouldn't pose a problem.
Would that be different? So for actually, it might be worth saying a little bit more about the AstraZeneca. So we talked a little bit so far about mRNA, and I've got other questions on this. But it might be helpful for people because they used a vector, didn't they? A virus taken from adenovirus. Yeah. And could you explain a little bit, like, what's the difference between using a vector compared to, we've got our sort of conventional live virus, and then we've got our vector and we've got mRNA. Could you describe a little bit?
Yeah, that's an important question. So, when DNA and RNA vaccines were being developed and in early trials of them, once they're inside cells, they can be very effective for producing the required proteins that are used in the vaccine and then creating the vaccine response. But one of the key problems is getting them into cells. If you just put the RNA or DNA molecule into the muscle and just give it a rub, you have very few cells that take up the DNA or RNA molecule, not enough to really create a significant vaccine response. So there are different methods to try and improve that, and one of them is that Moderna and Pfizer have done, which is to choose a lipid carrier to get the molecular cells. Another one, which is very effective, is take a virus, such as an adenovirus, and if you put your DNA construct into the virus, then the idea is the virus will inject it into the cells and create a much greater vaccine response. So inside that virus is really a small bit of DNA that contains components of the virus, the coronavirus, the spike protein, basically. So that once injected, it will create more and more copies of the spike protein and create a vaccine response. So it should be effective. But that's the reason for using the virus, is because it's a very good way of getting your vaccine candidate into cells.
Okay, no, that's helpful. So can you tell a little bit more about why one might be more effective than the other? So it seems in the research, actually, when you dig down in the data of the AstraZeneca one, that actually it's probably not as effective as first was communicated in the press. So what might be the reason why that one wouldn't be as effective compared to mRNA?
Yeah, I mean so. And it might turn out that other formulations will be very effective, but there are issues with using any other virus. This virus is not a replicating virus. This virus is just a particle that will inject the virus vaccine into your cells. So it's not going to sort of start dividing in your body and creating another infection. But when you give somebody an adenovirus vector, if that person's already had an infection of an adenovirus, although this is a chimp adenovirus, so it's less likely predisposed, they're going to develop an immune response, not just to whatever you put into the cells, but also to the adenovirus. And there are pros and cons to that. And one of the pros is that you want to switch on the immune system in order to create an active environment where a vaccine can really be more active. But the downside of that is you might eliminate the vaccine before it can do anything. So if you create a very massive immune response early on to the virus, it might not have a chance to really get going as a vaccine. That's one potential. Also, it's possible that the DNA vector. So because it uses a DNA vector inside the adenovirus, it has to get into the cells, has to get into the nucleus, and we have seen that RNA seems to be better at producing good amounts of the protein required for the virus in the past. So whereas it has to go in the nucleus, be processed, come out of the nucleus into an RNA molecule, the RNA molecule is then processed to a protein. So it might be to do with that. I haven't actually dug into specific reasons why that's that is proven less useful, but that's potential. Yeah.
Okay. The I don't know if you know much about it, but the Russian vaccine is also am I correct in stating that it's also a vector vaccine as well? Because I saw something recently about them pooling data or something, or they're going to combine the vaccines or something. There's something to do with that they're working together more closely than they have been previously. Could you explain a little bit about that?
Yeah. So I haven't looked into the results from the Sputnik virus. I did see that Pfizer was thinking of collaborating. Sorry, not Pfizer, AstraZeneca. Collaborating with the Sputnik vaccine. Now, let me just say a little bit why they might do that. So, one of the questions we have to answer with any vaccine is not only can it prevent you developing severe symptoms of disease, also can it prevent you from spreading these to somebody else, which is a really separate question. And when people develop an immune response to a natural infection, so when people get exposed to coronavirus in their lungs, you get an antibody response around the lung lining, which is different kind of antibody response you would get to that in your blood. You might have heard a lot about immunoglobulin G, which is the major antibody that you might find expressed in blood and muscle. But on your in your lung lining, it's another antibody called immunoglobulin A, which is you find in mucosal membranes. And we don't know yet whether any of the vaccines we have will produce an immunoglobulin A response. And that's, although it's possible, there still are some that I know do in trial, but whether the vaccines we've got coming out now, and whether that's important in preventing that early response to prevent you spreading with somebody else, or and the very early stoppage of the virus, basically. So one of the reasons why you might combine viruses is you might want to say, let's broaden the immune responses to the coronavirus. It's not just one form of immune response. Let's try and target in different directions and broaden the immune responses, because you really want to get an immune response that triggers both the cellular immune response and the antibody immune response and all the other parts of the body in a way that's not out of control. Yeah.
Okay, no, that's helpful. So I think what would be interesting for me to know would be, do you yourself have any concerns, that sort of genuine concerns about the safety of the vaccines themselves? So say the Pfizer and the AstraZeneca one, is there perhaps one in particular you would have more sort of reservations about taking yourself, or?
I don't really have any strong reservations towards either. I have few reservations towards the mRNA vaccine because it's such a simple vaccine design. Very little in it can really go wrong. But both vaccines, I mean, we've been quite a lot of work done on both viral vectors and DNA vaccines and RNA vaccines in the last decade, the last 20 years, really, in terms of DNA vaccines, looking at their safety and what they can do. And they have a very good safety record, at least in studies. So, but the RNA vaccine is so simple, I have no reservations of taking that.
And that's what I said. I wrote a Facebook post in November about that, about sort of how I'd have much given what I understood about the Moderna or the mRNA vaccines, that it just sounded really simple. And I've been aware of the technology for a while, because I know there's a while back. I know there's still phase 1 or maybe phase 2 trials now looking at cancer vaccines that Moderna have been developing using. And that's mRNA. So, and it's looking really positive as well. So I was kind of aware that the technology has demonstrably been shown to work previously. So if the choice is given to you, if someone said to you, Jocelyn, right, tomorrow you can have a vaccine and you can have the AstraZeneca one, or you can have the Pfizer one, which would you have?
Oh, I think the first shot would be probably the Pfizer vaccine, actually, because it looks more effective, and the mRNA vaccine is relatively simple. Yeah, I'm not saying I wouldn't take the other one. I probably would, but this one is like, yeah. I
mean, one of the difficulties is my understanding is the environment that they have to be held in. So the one you have to have, it's much, the AstraZeneca one is much cheaper and is much easier to be refrigerated compared to the. Are there any issues? I know some people have raised issues around the fact that because the Pfizer one has to be kept chilled, if there are any issues in terms of keeping it adequately cooled and things like that, could that radically affect the efficacy of the vaccine itself?
Okay, so let me explain why it has to be kept very cold. RNA as a molecule is quite unstable. In your body, you don't want to have RNA in your cells active all the time. So the body's very good at destroying it as soon as it's being used. There are mechanisms in place to digest the whole thing, to stop it being active when it's not needed to be. And because of the chemical structure of RNA, it's prone to degraded generally in the atmosphere as well. It used to be a joke in the labs, if you're working with RNA and you stare at it wrong, it'll degrade. So in order to prevent that happening, it has to be kept very cold. So if it's freeze thawed a lot, or if the temperature is increased and the molecule degrades, it'll be less effective. Yeah.
Okay. Okay, that's good. That's helpful. Thank you. I guess a few sort of. Oh, actually, no. I know what question I was going to ask. How long would you estimate that you think T cell immunity is last? Because I guess there's no. I guess no one can really know. But I mean, sort of theoretically, or if there's any precedent for it somewhere. What? Because I've heard sort of some people potentially saying it could be anything from just months, could be 6 months, which I think is unlikely. Or it could be 15 years or life, which again, I mean, I don't know. It could possibly, I don't know. What's your sort of?
It's early stages, but there are hints. So we have some hints and evidence. Now, of course, early in the year, in April, people are publishing articles saying that the antibody responses disappear or wane after 3 months, and that was causing a bit of some concern by people, which is understandable. Although antibody responses aren't the entire wing of the immune system. We have some hints from, first of all, from SARS, and also from, more recently, from MMR. So it's been noted that people who, at least in a small study, people who recovered from SARS have some resistance to COVID.
Right, that's interesting.
And that was in 2003. We also, a question was asked in the summer by some researchers investigating why it was that in countries that had MMR, fewer children were getting COVID than elsewhere. And it was, some studies from there strongly suggest that getting the MMR vaccine for some time afterwards have some resistance to I think I'm not sure yet, but we're thinking maybe up to 10 years for it to have some resistance to COVID. It's not as good as taking a COVID vaccine, but they're looking at, because of some argument will be there's some similarities, some of the proteins in the present MMR vaccine and some of the surface proteins in COVID that give you a little bit of cross resistance. So if both those studies, first of all, suggest that there's a possibility of long-term immunity. In addition to that, there's also studies done this year on COVID that look like you have responses that last at least 6 months. So T-cell responses and some antibody responses or B-cell responses last at least 6 months. So all that gives you hope for potential long-term immunity. There are other questions, of course, which is what happens if the virus mutates? And if the virus mutates beyond the limits of the vaccine, then of course you're going to lose immunity. Or if it recombines another virus, or so those would complicate issues. But as the virus
so would not the potentially the virus could mutate, but the spike protein could actually remain the same? Is that? Yeah,
exactly. So I mean, the virus could mutate. The spike protein could mutate in a way that doesn't make any difference to the vaccine, have a minor change, or it could mutate over time to be quite different. I mean, the spike protein could mutate to be quite different. I've not seen any. Well, potentially the mink recombination virus in Denmark. But other than that, I've not seen any suggestion that there's a strain of the virus at the present moment that will cause problems for the vaccine.
Okay, so I guess some of these things that the evidence seems to suggest that it's going to be long-term immunity with a vaccine. That's what you suspect.
It's optimistic in that respect. I mean, it might win after a while, but I'm optimistic that there'll be longer-term immunity. Yes.
So would this be something that we'd have to maybe get another one every 10 years or something like this?
That's the unanswered question. I mean, that's the question, first of all, with the AstraZeneca vaccine, would a booster help? And in terms of this, maybe there might be a requirement for a booster, yeah.
So there has been, have there been any documented cases? My understanding, there has been some documented cases of people getting COVID again.
I know one case.
One. Okay, yeah. I thought it was one or two cases, but yeah, maybe it's just one.
No, I mean more than one, but I just know one case. So,
in principle, my understanding is that says more about the individual. That's quite a unique. That would be unique, rather than. Go on.
Yeah. So the individual I know about was a person from Hong Kong who got a strain in Europe and then got a strain in Hong Kong, and now. Or the way around, actually got strain in Hong Kong, then I got strain in Europe. Now, the virus didn't make, so I don't know the history of his infection, but we do know that the first cases of coronavirus, COVID, were November last year. And there was in the press a noticeable, that there was a noticeable shift in the virus in December, January. So the virus shifted structure quite markedly. And that's a second structure that then threatened the world and caused lots of problems. So my first guess was he might have got the earlier strain. He certainly got, it certainly got two different strains of the virus that were different. So that's one of the reasons why I say mutation could change it. But since then, the virus appears to be relatively stable.
So when you say a strain, do you mean a mutation?
Yeah, sorry, mutation. Yeah.
Okay. So obviously, could you explain why there are several different strains? Because I know that there was, the strain in Europe is maybe slightly different in some way. I know in North America there are different strains. There is, I'm sure I've seen there are more than one strain of COVID. So if my understanding is correct, that's mutated the virus is mutated in some ways, but not in a way not on the spike protein. So we shouldn't be worried about any vaccine not.
Right. So RNA viruses tend to mutate over time, and it's one of the reasons they mutate is because every time they copy themselves, sometimes they don't copy themselves quite the way they should have done the first time. So you get a very, very slow change in mutation. You get occasionally the sequence changes by one or two nucleotides in the whole genome, and that causes slow change in the virus. Now, with the spike protein, which is the protein that the virus uses to attach to cells using what they call the AS2 receptor on cells, if it changes too much, it won't be able to bind to that receptor anymore. So there are limits within which it can change. It has to be able to change in a way that they still bind to receptor if it changes at all. So that puts some limits on its changes. So, but viruses do change naturally. So when you look around the world, you will see slightly different strains around the world of the virus, but ostensibly most of the virus is still functional and the same. Another way it can change, though this hasn't happened yet for the virus, thank goodness, is it can actually recombine with another virus. But that's unlikely event, but it's possible. That's what happens in terms of influenza.
Yeah, that sounds catastrophic. I don't want to think about that.
Yeah.
What are the chances of the spike protein actually mutating? Is there sort of previous examples of where a spike protein has changed on a virus, or was that are there parts of a virus that are more likely to mutate than others? Give us some hope about the spike protein not mutating.
Oh, I think spike protein has mutated. We've seen it mutate, but but the question is whether it mutates enough to make the vaccine effective.
Right.
And what's actually an advantage of the RNA vaccine is you can change it very quickly.
Right.
If the virus then changed enough, you go, oh, it's changed. You can very quickly roll out a new sequence of the RNA vaccine to change it. Yeah.
This is what I'm so fascinated about mRNA vaccine, because it just sounds like this is, it's been terrible, but for vaccine development, it's probably the best thing that could have happened. And I mean that just specific to the development. Yeah. I'm no way saying it's anything that's good about it, but it sounds like it's a real game changer in terms for the future, in terms of like if this really works, we in principle we should be able to develop mRNA vaccines for a number of at least viruses that don't mutate extremely quickly, like HIV. I mean, can an mRNA virus could be effective against those faster mutating viruses, or is that just out of the question, do you think?
That's a question. HIV mutates so quickly that it's a whole different level of difficulty.
Yeah.
I'm not saying it's impossible, but there are a lot of challenges to an HIV vaccine. Huge number of challenges to HIV vaccine, actually. I mean, people have been talking about developing one for a long time, and it's proven struggle. Yeah.
Yeah, it's one of the ones I keep seeing. I saw a meme a couple of days ago basically said, anti-vax meme saying that, we've had cancer for 100 years, we've got no vaccine. We've had trying to do everything we can for HIV since early 1980s, still don't have a vaccine. And yet COVID arises, and a few months later we've got a vaccine. Yeah. Which is silly, because I mean there's so much wrong with that. Like you said, HIV is not comparable to current.
HIV does two things that are problematic. One is every single time it divides or multiplies, it's mutating. It's got the proteins involved in copying itself don't work very well to its advantage, so it changes constantly, and that in itself is just a whole nightmare. The mechanism through which it binds to cells, the target for vaccines is just a difficult target to get into. It's proven, but there are drugs now that are very effective. So
antiretrovirals. I mean, you can basically keep your viral load so low that it's impossible to almost pass it on. I think it's impossible to pass it on. I think there've been some recent studies looking at that, which is great. I mean, so it's no longer it hasn't been a death sentence for over a decade now, at least. So I mean, that's pretty good. I mean, the cancer one I pointed out is that, the technology, there's already phase 1 trials for cancer vaccines from Moderna, the very company that's developed the COVID vaccine as well. So that's a pretty solid response to a pretty ignorant meme. Cancer
as a whole, again, quite different. Because cancers can be very individual to a person who's got this cancer they have. So you've got that technology called the CAR T technology now, which is you can spend $500,000 on and have immunotherapy to your specific cancer. But it's difficult treatment.
So what I want to try, we've talked quite a bit about how the vaccines work and RNA and things like that. I've got a few sort of questions more related to skepticism and sort of anti-vax sort of claims that I've heard recently. One is that that I've heard, again, lots of people have said this to me, that they're very worried about what's the sort of credibility that an mRNA vaccine can make you infertile. That's one I've heard recently. and what would be the mechanism? Because I couldn't understand the mechanism of how exactly that would work. But people have said, I don't even know where that came from, something that people thinking that you could be infertile.
Yeah, I came across that, not specifically RNA vaccine. I came across that, I think, originally in a post against, which company was it? Some other company, I can't remember.
Yeah.
There's no chance of this vaccine can make you infertile. I believe now Reuters, actually, the news agency, has done a really good job at, and I would look at some of the articles because they've done a brilliant job at finding the sources of some of these claims. And I believe the origins of that come from the fact that, I don't know, about 30 years ago, a group of Indian scientists were investigating vaccine treatments against testicular cancer, and they tried to target some male hormones as a way to attack that cancer, and that would of course cause potential infertility. But none of those components are in any of these vaccines. It's literally it's impossible. At least I can't think of any biological mechanism through which it can happen.
Yeah, but I just keep hearing these. They just seem to get they get wild.
Yeah.
I think one of the things that's really frustrating for me is I have good friends and family members and stuff that will listen, and you'll basically, all the videos start off the same. They'll have a doctor or scientist just laying out their list of credentials. I went to this this medical school. I've done this for this year. I've worked for this company. I've been a professor here for 2 decades and things like that. And then you think, oh, okay, well, maybe they've got something sensible to offer. Maybe this is, legitimate concerns. And then they just start saying stuff that's just crazy.
Yeah,
but it lends credibility, and that's the thing, is that historic. Well, I mean, even before the internet, but most of the information we get in terms of the knowledge that we get is from authorities. We can't be experts on everything. So, we defer to authorities. This person seems like they have the credentials of someone I should trust. So if my doctor says something, I trust my doctor that he has my best interest, he or her have my best interest at heart. And I can trust what they say. Yeah, all right, you have this wrong, you should take this medication. But where that goes wrong now is you have, there's such variation in humans and expertise that you can get doctors and scientists who have good credentials, but there are extremes. You've got, on one hand, you've got these people who are all very sensible, and these other people who are just kooks, but are intelligent. Because being against, being anti-vaccine, it doesn't mean you're not an intelligent person.
No,
but something has gone wrong in terms of the process and identifying and sifting information for what's reliable and not reliable. But the problem is the videos online allow people to present themselves as an authority and someone who should be trusted, but actually what they're saying is kooky.
Absolutely true. And I think my advice to anybody, is if you can look at the peer-reviewed literature. Some of it's going to be difficult and technical, but many of the medical journals are sort of British Medical Journal, The Lancet, Uni Journal of Medicine, are a little easier to read. At least you read the abstract, the summary. There have been a number of reports in the scientific media about the origins of misinformation, and there's been some concern that even states bodies responsible for some of it. Certainly, there's been a lot on Facebook, and there's been lots on some of these charities. And it is sad. It is very sad to see people that have lists of credentials that then say things that you go, what? And I've seen some of the tendencies that I've seen are to use a lot of jargon, because using jargon can cover up, or can scare you, or can cover up the fact that what you're saying is nonsense. So I've seen language of exosomes, PCR, I don't know, DNA mutations, those sort of language. And check out, if the peer-reviewed journals are too difficult, check out Reuters, because Reuters are a good news agency, and they do make a lot of effort to try and sift through it, or the BBC, or whichever credible news agency you find. I am concerned also that there's a lot of distrust generally of mainstream media or of media outlets, and I know some reasons why, but at the same time, I think we need to be careful. And also, I guess personal stories sell. So if you do see a case, somebody who's reacted very badly to something, that story sells a very strong message, whereas leading statistics doesn't.
Yeah. And the thing is, there's always been, and there will always be people who are, incredibly intelligent, and will purport, things that are crazy. And so that's just the normal spectrum of any profession. You get cracks like Andrew Wakefield, who fudged data, and caused so much damage, even to this day. Has caused so much damage to the world through through a study that's basically falsified, and scared a lot of people into giving their children vaccines. So it's
Andrew did a staggering amount of damage, and still does to this day, from what was a paper published that was utterly false, made up. However, the two positive things came out of that. That it did make people think very carefully once again about what components are in vaccines and whether they should be reconsidered. So while I get very frustrated at what happened in the aftermath of Andrew Wakefield's fraud, really, to be positive about it, it did look at social reassessment of vaccine components. So vaccines, most vaccines, again, this might sell the RNA vaccine slightly. Most vaccines require not just the proteins that you want to use in the vaccine, they also require something else that was called an adjuvant, something that will boost the immune system more generally. And so in the past, they've used things like alum hydroxide and other components. And mRNA vaccine doesn't require that, because mRNA itself can stimulate an immune response. There's receptors in the surface of cells that will react to the presence of some forms of RNA. So it is in itself its own adjuvant. It's not a benefit.
I think this is the thing that's so frustrating, because actually there's something really helpful about the skepticism, about being skeptical and raising concerns. Absolutely, we should raise concerns and try and bring about improvements. Like you said, if there are excipients or other things that could be potentially harmful, or there are better things that could be used in instead of them, then we should try and facilitate and encourage change in the pharmaceutical industry. But a few other things. I mean, again, this one video sticks in my mind. Again, I can't remember the name of the doctor. But then, this list of credentials, and then started talking about 5G. I'm really interested. Is where did the 5? Do you know where the origins of 5G?
I have no idea. I know that people are concerned generally about 5G. There's been a number of what I'm going to call conspiracy theories about 5G for at least a year now, and people taking down 5G. And the concern that 5G in itself might be a mutagen that might damage your DNA or RNA, but it's not actually the frequency that it's in. It's nowhere near the area that will be damaging to the human body. So I don't know where 5G coronavirus came from.
But again, I mean, with 5G, again, that's where people should be, if there are concerns, because sometimes I think, if someone just suddenly you look outdoors and someone's put a 5G, 30-foot hole outside your house, I think it's perfectly normal to have concerns. Is that going to have, health impact on me and my family and things like that? Because we haven't all done the research, and we know that there's historical precedent for things that were intended for good that actually turned out to be harmful. So I think it's always good to have those reservations.
This is more my experience than that's not the truth necessarily, but many of the better scientists, the ones that just stay in the background, don't say anything. They just, because they like to look at the data and weigh it up. And sometimes people at the front are saying the polemical stuff are the ones that get the most voice. And those that are more reasonable or say, well, actually, it's not like that, are the people that don't like being in public and stay in the background.
Yeah. Yeah, I mean, another one that a really popular one is, I think, that I've seen is, does mRNA vaccine actually change your DNA? That's another one that I've heard quite a lot
that's
of
not a
times.
question. No, it can't. It can't. Because RNA is chemically different to DNA, it's actually impossible for RNA to get into your DNA. It can't do it. It's impossible. It's impossible chemically.
See, I think what people mean, I think there's some confusion. So it doesn't, it's clear that the mechanism of RNA, it can't affect DNA. It doesn't change your DNA. I think what people mean is it's changing the body. And that's fine. Maybe I'm trying to be as charitable as I can. I think maybe that's what people mean is it's changing the body in a way. So it's not changing your DNA, but that is the nature of all vaccines and in a way, because if they if you develop T cell immunity to a disease, then that has changed your body, but it hasn't changed your DNA. You're fundamentally the same person with the same 46 chromosomes that you had before. That's
a good question. But it's think of it a little differently. Think if you naturally get the coronavirus infection, that the coronavirus binds to your cells, injects its RNA into your cells, it replicates without the cycle of the virus multiplying inside your cells. That's much more. That's having much more effect in terms of its biology than just putting one component of it into your cells. So into cells that are run, it'll be in the muscles, so it'll be in a limited area. Whereas coronavirus natural infection could be quite covered, because the coronavirus can affect not just cells of the lung, it can affect the cells of the brain, kidneys, liver. Quite widespread infection if it gets serious.
Yeah, I think that's a lot of people fail to think of, especially when people are maybe underplay the seriousness of COVID. Is that I know what we've learned recently, and I think we've even since August or September that that there can be variance in the severity that was observed because there is a sort of genetic component to it. So certain people with certain genes are much more likely to experience a more severe disease process compared to many others. So for some people, they get a cold, a temperature, and a headache, and other people are in intensive care and renal failure and respiratory failure and perhaps end up dying. So how would that work? Why would do you know anything about that? Why would some people get a more severe response to others, or was that?
Well, so there are a number of reasons why people might have a more severe outcome from COVID. The most simple one is how much COVID they got in the first place. So if they got one virus particle in themselves, it'll create an immune response and create an infection that will take some time for it to get going, and then the body will probably kill it off before it can do anything, and then you've already got an immune response. If you've got a large dose of it, let's say you're working in a hospital and you're continually meeting patients who are breathing in your face, then that's one factor in itself that could result in a poor outcome. Another factor would be your biology. So we know that, or actually your diet as well. So so whether your diet is giving you a proper balance of nutrients that keep your immune system healthy. So the vitamin D's and zinc, both very important in the natural diet. It could be because the receptor that the virus binds to in your body, the ACE2 receptor, everyone's got their own little form of it, and some forms make it easier for the virus against your body than others, so that can affect it. Again, going back to vitamin D, the receptors, vitamin D receptor in your body could have different forms, which can make it more or less likely for you to get it. And also, there's your immune system itself has different variations in it, and the variation in the immune system, which can be actually across populations, can also change how you might react to the virus. And we know age is a factor. People that are older, particularly over 60, 70 years old, are more likely to have a severe outcome. Yeah.
I'm sure I read something today. I was reading a Nature paper about type 1 interferon and how you know, if you have a genetic mutation that related to that you have, that you're in the sort of, you're very likely to have one of the sort of bad outcome with
Absolutely,
COVID.
absolutely, yeah. And that type of interferon, so the interferon response is a natural antiviral response your body has. So if you've got mutations in interferon genes or changes in interferon genes, they can seriously affect your ability to react to a viral infection. Yeah.
Okay, another one I've heard quite a lot of, and again, someone who's sort of quite active in sort of pro-life philosophy and things. I'm sort of thought about these things before, but a lot of people are concerned about not wanting a vaccine because of the involvement of fetal cells, so specifically sort of HEK293 cell line. Now, I've heard things from people, a lot of people. There's groups of people that think that there are fetal cells in the actual vaccine itself, which is absolutely not true. And then there's others who are aware that in many of the vaccines, if not all, most or all, have used a fetal cell line in development. So could you tell a little bit about, you know, why do we use fetal cells in vaccine development, and why do some use them but some don't? It would be interesting to get a bit of background. Talk about the ethics of that a little bit, perhaps.
Yeah. I mean, so 293 cells, as are probably become more widely known, originate from cells taken from a fetus in the early 70s, and those cells were taken and grown in cell culture. So once the fetus was aborted fetus, cells were grown in cell culture, and then they were transformed, which means they were made immortal by various means in the lab, basically. Now, and since then they've been grown as immortal cells. Now, the reason why they're extremely widely used, that cell line is very, very widely used in biomedical science. And in fact, although I couldn't be certain, I would say most cell biologists have used that cell line, even if they're not aware of the origins of the cell line. When many people, I'm not sure that all cell biologists will be aware of the origin of the cell line. They'll just turn up on their desk at 293 cell line, and they'll go, oh, okay. Or it might have a different name because sometimes they've got brand names because the 293 cell line itself has been modified to do other things. So some cell lines will be called a particular brand name, and you go, oh, I'll use that cell line and not know its origins. It's one of the reasons it's very widely used is that it's quite a well-characterized cell line. The cells themselves, it's very easy to get genetic material into them. And one of the difficulties you have in working in cells in the lab is getting your if you're going to do tests on genes, it's getting the gene into the cell in the first place. And the 293 cell line is a cell line that's very easy to get DNA into or RNA into, whatever. So that makes them just an easy, and they're quite big cells, so it makes it quite nice candidate to work with, generally speaking, and do initial tests in them, and they're easy to grow. On top of that, there've been a number of people who've modified the cell line to produce for research into viral vectors, whether they're retroviral vectors or, in this case, adenovirus vectors. So cell lines are modified in a way that some of the components of the virus are produced in the cells themselves, so that any particles of virus coming out of the cells won't be able to divide. They won't be active viruses because they're assembled inside the cell, and it requires the cell to be there, the 293 cells to be there for the components of the virus to come together. Without the cell line there, nothing can happen. So the cell lines, because it was derived from cells from aborted fetus, the cells now don't really resemble the kidney cells they originated from. They're just a kind of amorphic cell line. And for that reason, I couldn't be certain every single vaccine manufacturer has used them, but I think a lot of studies that have been done in COVID and cell lines have used this cell line. Looking at it, people initially looked to see what cells COVID would infect actually, and that was one of the cell lines that was chosen. And then people took that cell line and gave it, say, the S2 receptor, so that it would be more easier to infect that cell line. So it's been used very widely in development. Yeah.
So are there alternatives that could be used? And if there are, why aren't they widely used? Because I know some vaccines haven't used them. Not necessarily. I'm not sure about the COVID ones. Although did I read something about a Japanese one didn't use fetal cells in development, but I'm not sure. I'm sure there's at least one COVID one, but sort of vaccines more generally. Why why don't they use alternatives if there
Well,
are?
so cell lines come from usually cell lines that are used in labs have to be immortalized cell lines. So they often come from tumors. They come from occasionally come from an aborted fetus. There are a number of options for cell lines. You could use primary cells, the cells taken straight from someone's body, but they don't survive very long. You can only use them so far, and then they quickly lose their functionality. So it makes them difficult to do long-term experiments with and make this kind of reproducible. There are a few other cell lines that have been used, and but I have to go and look at what cell lines would be actually useful in terms of can be infected by COVID and can be used for studies in terms of whether proteins are expressed in COVID and response to it. So, but it's one of the most it's used because it's one of the most convenient cell lines to use. I think that's basically the reason.
So, I mean, in reality, they're not going to be. It sounds like they're going to be continued to be used for the foreseeable future. With that,
I think 2 cell lines will be used very widely around the world for the foreseeable future. Yes. Yeah.
Okay, so you said that you would be willing to have the vaccine. So how do we kind of ethically navigate the fact that aborted fetal cells are used in its development process, yet you would accept the vaccine?
Yes, and I would, and of course, it's probably when I'm a Christian. So, what my position is in terms of taking the vaccine developed from cells originating in aborted fetus. I mean, I think the fact that abortion took place is a tragedy. However, from my perspective, that's not something that I can change whether or not I have the vaccine. Whether or not now whether the cells were the vaccine produced in the cells, or whether those cells were used during the development of the vaccine, to me are morally equivalent, and because they were used at some point in development. But I cannot go back, and I'm not going to be condoning use of abortion, nor am I going to, but nor can I change that going forward. Nor can I prevent that happen, nor can I prevent future abortions happening by my actions. So I don't have an ethical problem taking this vaccine.
Yeah, no, it's good. I mean, I've always thought that the analogy would be if, say, a child was killed for some reason, by a parent, but they were, murdered by someone, but they were an organ donor.
Yeah.
The mere fact that you accepted an organ from someone, I know in the case someone's, it gets a bit murky maybe when you start talking about let's say we're in an opt-out system of organ donation, and someone is murdered, and by receiving that organ donation from someone, the fact that you accept it is not tarnished by the fact that you're not condoning the murder of that person by accepting being the recipient of an organ from someone, in the same way that by utilizing the cells from an aborted fetus, you're in no way condoning abortion. Abortion is wrong. But we live in a world where in many countries it is legal. In the country those cells were taken, that abortion happened was legal. And those cells were taken, and there is significant moral proximity. We're not morally proximal to that act. There's nothing, like you said, there's nothing we can do to change the evil that occurred. But we are, that good can come from that death. There was an unjust death, but good can come out of it, and the world in many ways is with the benefactors of that, but in no way condoning no abortion. And as I said, in the same way that accepting an organ from someone who was murdered doesn't mean you're condoning the murder of that person, but you're benefiting from an unjust death.
Yes.
That's kind of what I think about it. I'm not sure
and I thought when the issue came up, I also thought more widely about what knowledge we have gained and made use of situations that in the past have been morally horrific. Whether that was during the war and human experimentation that was done during that period or afterwards. or before, in some cases, looking at medical effects on people who were prisoners, or whichever. And so those issues, and these are important issues, and issues that I think need to be brought up. I think it's right we have those voices, because we have to continue to question these issues. But I think in this particular case, I don't feel any more qualms taking the vaccine.
Yeah, I think it's a good question to raise. Like, here's the thing: like, some people are uncomfortable talking about it, but I just think no, it's good. We need to talk about it, because it's a good question. Why are we using aborted fetal cells? And we have to talk about it, because I think when we don't, if it's just an instantaneous sort of impulsive response to it, we'll be like, "Wow, well, it used aborted fetus cells. I'm against abortion, so I can't do that." But actually, we have to think about these questions more deeply, and reasonably, and think actually, that death was bad, but that's not enough to justify not utilizing vaccines. And I think, like you said, I mean, historically, what has sped up medical and scientific development exponentially has always been war. It's always been death and violence, and we've all benefited. Each and every single one of us has benefited from from war and violence. My own areas, in the operating and perioperative care, and surgery. The main driver of surgical advancement and medical advancements has always been war and conflict. So it's the part of being human is, especially morally, it's just everything is meshed with good and evil. It's so hard to unattach yourself from and try and live unattached from past moral evils and things like that, and injustice and things like that. But I guess we've got a few more questions before we finish up. I was hoping we get some more questions. It's a bit light on the questions so far. I think I've asked the ones that have come up. I was hoping we'd have people who were there
was a question, wasn't it? At least I saw in the chat about low-income countries and what's going to happen about low-income countries.
Oh yeah, that'll be a good one. Yeah. So what how is it fair that the West again benefits from vaccine development, and what about sort of
So I mean, the different vaccine candidates are now being produced, and the number, and as we mentioned earlier, the RNA vaccine has to be kept at a very low temperature, 97 degrees, and that makes that vaccine more difficult logistically to transport to countries where there are not good refrigeration techniques or freezing techniques. That doesn't mean the end of the story. So, and somebody asked, what should church be doing? And I would ask further than that, what should Christians be doing? Not just what should Christians be saying, but what should we as Christians be doing to help the situation? Now, there are a number of international organizations that have, WHO being one, but others who have committed to huge number of vaccine doses to try and help, of different sources, to try and help countries, the low middle income countries, to get vaccines, to supply vaccines. And also we have to ask the question, how can we improve logistics so that those countries can receive, have an option of receiving different vaccines? So the AstraZeneca vaccine is more stable. It can be kept in the fridge. So that's easier to transport, but of course we have to optimize its efficacy. And the Moderna and Pfizer vaccines have to be kept very cold, so there's a need probably to find means which they can be distributed cold and dry or whatever. But so Christians can get very involved with this. I think it's important, I think, not just to shout from the edges. It's very easy to criticize others for what they're not doing. But I think what's important is to get involved with helping, whether you then say, okay, I'm concerned about these issues. How about I give money towards an organization that is actually helping to provide vaccines? The Bill Gates Trust, I think they've put in, Bill and Melinda Gates Foundation, overall this year put in 1.75 billion towards developing the COVID response, including money specifically towards giving vaccines to the third world.
Yeah, I think it's the COVAX initiative, isn't it? I think
Yeah.
involves maybe like 80 countries who are providing, funding for vaccines for another 70 odd countries, I think.
Correct, yeah.
I think, yeah. So yeah, it seems like actually, I mean, it doesn't seem unfair for the fact that it's the West largely that has funded the research. So obviously, I think it's no one would understand it as being unjust. I think for us to be the initial benefactors of that, but absolutely, we need to get the vaccines out to countries that are not so fortunate economically and things.
I was thinking back to development of HIV therapy. So in the 1997, I think the year was, when combinatorial HIV therapy came in, palliative viral therapy was first distributed around the world. One of the questions then the world was asking was, well, these drugs are very expensive. You're spending $25,000 a year on treatment for one individual to receive antiretroviral therapy. How is it possible that those who are suffering from HIV in Botswana or Malawi or other countries can possibly afford those drugs? So there was a lot of effort putting in by various activists and to try and say, can those pharmaceutical companies offset the costs? So the cost of antiretroviral therapy is much cheaper in Africa, actually. The cost per patient is considerably reduced to try and get the drugs out to those who need it. And the West, the prices are a bit higher in order to try and offset the costs. So there's, I think, also need to think about, are you willing to sacrifice, and we should be willing to sacrifice some of the cost for us so that those who cannot get it can receive the vaccines.
Absolutely, yeah. I mean, it sounds like the COVAX and a number of things that are ongoing as well. I think that it's as good as it could be. I think it looks really positive in terms for a lot of countries around the world. So yeah, I think a lot is being done. I don't think too much more could be done really in terms of funding and agreement from a lot of countries with the resources to fund those. And another issue I've heard is a general sort of claim is that people about vaccines is that people shouldn't intervene with the sort of normal human processes, and so we should just let the virus take its course, and just let the body is best. The immune system is great at what it does. Actually, we don't need to try and improve it by the use of a vaccine. So how would you respond to a claim like that?
Well, I mean, I think we're very fortunate actually to live in the age that we do live in, because in the last century or so, we haven't had to face some of the pandemics that have faced the world over the centuries. Smallpox has been eradicated. The incidence of measles is greatly reduced. The Black Death no longer seems to affect many countries. So I think that takes off some of the pressure from us. But going back, looking at just the Bible, we know that Jesus himself intervened to heal people who had natural affections, as did his disciples, and they were encouraged to do that by Jesus. And one of his disciples, Luke, was a doctor, and you see no criticism of his work throughout the Gospels. So I think there's biblical precedent for medical intervention for people that are suffering from natural ills, for this fallen world that we live in, really. And I do think that we need to think about how much blessed we are because of the rise of modern medicine in the last century. We faced so little of the diseases that were affecting people just over a century ago. Even TB, in the West at least, TB is greatly receded, although worldwide it's still an issue.
And cholera as well, not that long ago. Cholera, some of the plagues, you read about some of the horrendous stuff, you get the bubonic plague would get to a town or a village somewhere and it would kill 75% of the people that live there. Absolutely savage, the stress that you must live under, the fear that people would live in. It's really hard to imagine 75% of the people dying. We really, like you said, we really are. I don't think we always appreciate the times that we live in and the technology that we have, and the speed of the vaccine. COVID vaccine is really, a fantastic example of that, of how we're, able to do that so effectively and so quickly.
Actually, if we're also lucky that while COVID is a serious illness, it wasn't SARS, or it wasn't MERS that hit the world, because SARS had a much higher mortality rate.
Yeah.
And we would have struggled to respond to it at that time so quickly as we've been able to respond to COVID now.
I think the good thing about SARS was that obviously I think people did become, by the time people were actually infectious, they were really ill, so they tended not to be exactly around other people. I mean, going back to the sort of intervening in normal human processes. I mean, as I said, we do that all the time. You know, we wear shoes and clothes to facilitate homeostasis and normothermia and things like that. So we're already doing it. We all the time. So I do have a few more questions, but I've got a question that's come up. So Richard Conway from Facebook has said, has COVID-19 actually been isolated yet to prove its existence? Yes,
in fact, it was isolated by two Chinese researchers soon after the disease appeared in November, December time. I think the first report was the 8th of December, likely, but it's going to go back earlier than that.
You can by January.
If you want to go online, if you want to search COVID sequence, you'll find that there are thousands and thousands of the DNA sequences that have been published of each individual virus because people are trying to track the strain. The knowledge is, the information is public in the public realm. If you want to say academic database for that, you can check PubMed, PubMed, but that might be a little academic. But we've isolated it and we've photographed it.
Yeah. So yeah, there's absolutely no doubt, zero doubt that coronavirus, I think COVID-19 exists. It's been isolated very early, and that's why companies like Moderna were able to. I think it was literally within a few days. Because of the nature of mRNA technology, I think even within a few days, they had a vaccine sort of pretty much there. In many ways. Which is great. It just shows the speed and effectiveness of that technology and the potential that it holds.
And one of the reasons why we were able to isolate so quickly actually was because those researchers in China who foresaw the disease recognized it as a coronavirus type infection, and already had the tools available to get at the genome, basically.
And that's a really important factor as well that a lot of people forget. It's just isolating a virus. It was really, obviously you're going to know a lot more about this, but it was really by accident that we were, when the HIV virus was identified in sort of early 80s, we didn't even know retroviruses exist. Like theoretically, my understanding of the discoverers of DNA didn't think it was even possible for there to be an RNA virus to exist in humans. I know I found there was a cancer causing, I think it was a leukemia causing one in chickens, wasn't it? I think
HLV,
So you'll know more about this. I'm just dabbling.
So it took years of work to isolate the cause of HIV in a number of labs around the world. One of the reasons HIV was such a difficult problem was that the incubation time for symptoms is normally 8 to 10 years, it's a very slow growing virus. It was very, very hard to find the virus particle. It took a lot of work by a lot of quite hard working scientists at the time. And these days it's very easy because we know what it looks like. But they
were ridiculed, weren't they? I remember, I'm sure was reading somewhere about how the scientists who even suggested there could be a retrovirus were ridiculed. They wouldn't get their papers published. There's a really good episode on that for anyone who's interested on Malcolm Gladwell's podcast. I can't remember, something about the Virus Club or something like that. It's a revisionist history, but you probably really enjoy it. A really interesting episode on the Virus Club or something like that. In there, I guess a few more questions, unless we've got any more coming through in the next few minutes.
The one thing about HIV is one of the early concerns was people were saying it's toxin. It's not caused by virus. It's caused by toxicosis. By some of the drugs, as AZT or something else that given. But these are good questions, but they're all false leads. And again, in that time, it was spun out into a conspiracy theory in some of the news outlets. So we need to be careful about we step back a bit and look at.
I remember what the Obscure Virus Club. That's what it's called.
That's good.
It's really excellent. It really is excellent. We've got another question that's come up. So. it has, then why, when requested to, did the government not provide proof or documentation of it? So I think this is in relation to the coronavirus actually being isolated to prove its existence. So we said that actually it was published in the late, December, January. So if that's the case, what you're saying is true. Then when requested, did the government not provide proof, documentation of it? I'm not quite sure.
I'm not aware the government, I mean, first of all, I'm not entirely sure it's the responsibility of government to provide documentation. Maybe the media should be broadcasting what information is available in the scientific press. I mean, the questions are being asked very early on in January and February, were having found the sequence of the virus and the virus, what its relationship was to other natural viruses in bats and other animals at that time. So, and it's certainly the media should have been responsible in publishing that, but they have. They published the fact that it was coronavirus. I don't see any government cover up in this.
No, I'm not aware. I mean, it's an interesting question because this is all freely available. You can actually look at the sequence of the COVID-19 virus online. It's freely available, open access on peer-reviewed journals, and it's been, like Jocelyn's already said, it's you can look at pictures of it. It's there. There's no debate about its existence. And as you said, it's not the government's. The government is not hiding the existence of it. As we said, it's freely available. So why is there a big question? Just generally, I know we touched on it earlier, but why is there so much misinformation about vaccines? What is it about vaccines that is something that we talked about is so good in terms of we don't have to worry about smallpox anymore? We've nearly eradicated polio. And numerous other things in the pipeline, cancer vaccines, as we said, Moderna in phase 1 research into that, and numerous vaccines that are available. So why is there so much pushback, do you think, and misinformation against vaccines about something that we should be celebrating? Everyone should be celebrating, and yet there's a large portion of people who are not just skeptical, because that skepticism is good, there's healthy skepticism, but overly have an unhealthy skepticism against vaccines.
I would imagine it's in part because of medical tragedies in the past. So we know everyone's very aware of the thalidomide tragedy that happened and the consequences of that. And then on the top of that, you've got the Andrew Wakefield's conspiracy, really, in the 90s. And
could you explain a bit about that, just for people, about Andrew Wakefield and exactly why we shouldn't? I don't know if you're happy to. Why shouldn't we trust the study he said? I mean, it was retracted, and obviously it did a lot of damage, and we're still dealing with that now. But why shouldn't we trust his research? What was wrong with it?
So he published case studies. I think it was 12 children whom he argued in the paper in the Lancet had developed symptoms of autism as a result of having the MMR vaccine. Now, in the wake of the publication of that article, so it was a peer-reviewed article, originally accepted, it was discovered that, first of all, he had a competing interest, that it appears that he was being funded by a legal firm to find cases against vaccines. In addition to that, that there was considerable concern over the fact that in the paper itself, the data were made up, that the case studies the children's symptoms did not reflect the truth. I think a journalist, of course, John Deere, I've forgotten his name, the journalist, but a journalist did a very good job in following up and interviewing individuals in that study. And more importantly, I think, is that it did stimulate people to look more widely into the vaccine. And large-scale studies were undertaken to show that, and showed no link whatsoever between autism and the MMR vaccine. And there's no one's been to repeat those data. So even if the data in that original paper had not been retracted, they were never reproduced. And that's one of the questions you have to ask. Whenever you see a scientific paper, and whatever that subject is, it's very important that those data are reproduced. It's one of the first things you really have to do to show that that's a consistent effect and not just something that you saw, whether because you did something wrong in the experiment, or whether there's something abnormal about what you did, or whether you made it up. So I mean, the studies now been done on tens and tens of thousands of individuals who had MMR, or hundreds of thousands of individuals who have had the MMR vaccine, and show no link whatsoever between that and autism. But the damage was done. And I remember watching an interview, or listening to an interview on the radio once, between a woman who was very concerned that her child had developed autism as a result of having the vaccine, and the clinician who had looked at the wider studies. Now, on the radio interview, it can seem as though those two sides are equally weighted, because the woman's telling a very concerning story, and it's tragic. Whatever happened to her child, and I don't know, because I don't know the case at all, is a tragedy. But that doesn't necessarily weigh up against the clinician saying, well, actually, we looked at the evidence, and here's the wider evidence, and we don't find any link. Whatever the cause of your child's disease, it doesn't necessarily link it to the MMR vaccine. So it is stories about, heartfelt stories about people suffering are very tragic, and they sell widely, but it doesn't necessarily show the whole picture. So I think we should be very skeptical of those data. But I think it had a terrible, terrible effect on any vaccines subsequent to that, and has been used. It may have been used even by people who want to sell Discord.
Yeah, I mean, the 12 case studies, I mean, the thing about case studies is case studies are great in many ways, because they're usually the first indicator that there might be a problem. Yeah. And there's been examples that, I mean, even with HIV was picked up with case studies, you know, initially doctors identifying case studies along, I think it was the West Coast and elsewhere. Yeah. And there's been other cases where issues with, you know, drugs that have been, you know, there's been sort of case studies about, what was, it's completely lost my mind. The one, the anti-sickness drug, morning sickness drug. Thalidomide, that's okay. Yeah, sorry, yeah. I completely lost it. But thalidomide was another one that was picked up with case studies. Yeah, you know, so I'm not against case studies, and I'm sure that's not what you're saying. But you know, in this case, you know, it wasn't representative, and I think with a case study, then the good thing is it pushes you to do more research. And there's been enormous studies, like you said, and numerous systematic reviews that have shown that there is no link. So it's dead in the water. There is no link between MMR.
I think to the those who are concerned, I think sometimes the medical data doesn't get out beyond specialists.
Yeah.
So it's not, we see the data, we see the result, but it doesn't get out into the media, because it doesn't sell. It's a scandal. It's not a scandal, so it doesn't sell so easily into the public media. But the information probably needs to be out more widely so people can see the bigger picture.
Yeah. So another question from Richard Conway again from Facebook. Good question. So he says, forgive me, but with the amount of censorship and fact checkers, it's difficult to filter out truths. And I think that it's really difficult. I think the problem is, really good question, Richard. And I don't know how we're going to navigate it, because there's so many people saying different things. We have to get, it's a skill in itself, knowing which sources to trust, because like you said, like we depend on a lot of the knowledge that we have from experts, you know, from authorities. We defer to authorities. And, you know, if you've got a scientist or a doctor who's very, you know, sounds very prestigious but is saying things that are untrue, how, you know, it's very difficult to know that. I mean, often a good kind of rule is that, you know, if someone is in a minority view, that's often not in itself, just because, you know, you can get people who hold minority views and actually they turn out to be vindicated in the long term. So that, but as a general rule, you know, if you've got, you know, 100, you know, out of 100 scientists, you've got 99 saying this is the case, and you've got one saying this is not the case, something else, then the fact there's only one out of those people saying that would lend you to have some skepticism towards that one person. It doesn't mean what they're saying is not true, but we should have a healthy skepticism towards that person. But what's happened? I don't know why that's happened, but some people can put the trust in the one person rather than the 99. Yeah. Well, that is the case. I think when it comes to
people see the fear and they think, well, that could be me. I mean, you see the potential side in case of the allergic responses in these two individuals that got the COVID vaccine, and you see that could be me. So that immediately your fear response goes off, and one of the dangers, of course, of looking at even peer-reviewed literature, is that there's so many peer-reviewed articles on COVID, and there's a lot of papers out there, almost thousands now, that you could find that one paper that's giving some potential danger and read that as if that's the whole picture. So I think if I've got any advice at all, it'd be look at Reuters first of all, because their news edge is a little bit distant from some of it, and they will look widely. If you're going to look at a peer-reviewed article, try and look at review articles, because they tend to give a step back and try to bring a lot of the data together into one article, or talk to people who are working in the field, working in the area. If you get the opportunity, I don't know. It's not easy. I think if you wanted to go on Google and just Google it, you have to read quite widely to see the whole picture.
Yeah, I mean, things that are usually again, not the blogs and things like that, but websites that are not things that are not peer reviewed, like threads in forums, can be great sources of information, but they're not. They're often full of a lot of unreliable information as well. So I think, like you said, when it comes to questions of medicine and science, the best thing to look at is a systematic review. Because if you think about evidence as a kind of pyramid, they talk about a hierarchy of evidence. So at the bottom of that hierarchy, of that pyramid, you would have something like a case study, an individual case that might just look at one case. But when you look at a systematic review collates all of the research and the studies that have been done on answering one particular question, and summarizes those studies. It looks at the integrity of each of those studies and will rate them, and will give you an answer based on all the existing research. So if you and often you can just get the information just from reading the abstract. You don't have to read the study. You just look at the abstract and look at the conclusion, and that information is going to be better than probably almost anything that you can find. And I mean, the difficulty, one of the problems with sort of academia is that it's often behind a paywall, the whole study, but abstracts are usually available. So I think, like if you really want to look into a question, especially stuff around COVID and vaccines, is look through something like PubMed and look, or Google Scholar, and read the abstracts of systematic reviews. So that's about as good
We're very lucky, actually. A lot of the COVID publications have been
open access.
So open access, which makes it brilliant this year, actually.
And just so you confirm, because there's some skepticism around you that you have, you don't work for Cambridge University, do you?
No, I don't. No, I don't.
What was you studied at Cambridge University?
I did work at Cambridge University for a while. I did a postdoc there 10 years ago. I did study theology at Cambridge University, but I do no longer work for Cambridge University now.
So you're not benefiting financially from anything.
I don't benefit at all. No, I have no link to anything to do with the vaccine. I'm only talking because my experience in immunology just made me concerned about some of the conspiracies that were arising.
Yeah. And I asked you as well. I think that's the main thing, as I asked you. Okay, another question just come in. Thank you, Richard, for these questions. They are helpful. Okay, so why are the governments of the world pushing this vaccine so hard for something that's not exactly deadly to most people?
Well, so it's true. Most people get a lot of mild infection, but a significant number don't. A significant number end up, I think it's 10% roughly, end up in severe hospital care in the UK. And it is, I think the current mortality rate suggests about 3 times the mortality rate of influenza, which is quite significant. We do vaccinate against influenza, remember, every year. It's recommended people that are at high risk get the influenza vaccine, because influenza in itself can be quite dangerous. And this vaccine, so there are a number of concerns. One is you're benefiting not only potentially yourself, but also the community around you, preventing those at a high risk from developing severe disease. And I think also there's a question of if the virus is allowed to freely roam in the environment without any attempt made to prevent it spreading, it gives it more chance for it to mutate, more chance for it to become potentially more severe. I'm not saying it will, but it's just not a good virus to have kicking around in the environment, in my opinion. And bear in mind that 10% of the population going to hospital care is a lot of people. And that's quite a strain. If that happened at one time, it's a big strain on a health service anywhere in the world.
I think one of the things as well is if we theoretically we thought about we did nothing. So we just said, look, everyone go and live how you were before.
Yeah.
And we'll do nothing. The surge you would get from that in hospitals,
oh, horrific,
it would be horrific. And that is a fact. That is a fact. It's horrible. I don't want to be at home. I don't want to be at home doing it. I haven't seen anyone for months, other than my kids and my wife. I don't want to be doing it. But you know, there is benefit in doing that and restricting contact with people. If, as I said, if you did nothing, not only do you have to worry about the deaths of people from COVID, which fortunately we're getting much better at treating. You know, during one of the things about the lockdown that was beneficial is it basically meant that hospitals were not absolutely flooded with people with COVID, because we didn't know how to treat the disease very well. You know, there's so many things we learned. If you look at dexamethasone, which we use all the time, cuts the rate of death, I think, by 33% in those with the most serious COVID. That's amazing. That's great. And we were only able to discover that because we were given we had the time to do that, and the lives that have been saved like that will be hundreds if not thousands of lives will be saved by that development.
At the early stages of COVID, I mean, what we had experience of SARS, and aftermath of SARS, people did actually try and mold antiviral drugs in tissue culture against SARS to see if any would be effective. And so the early surge of drugs looking at effectiveness, efficacy of drugs in humans, were trying some of those drugs already tried in SARS. That's where hydroxychloroquine came from, because that had been already tried in SARS. And looking at, so there was but we didn't know anything. We were making educated guesses based on our past experience, but it's taken a long time to really kick up the medical care. And if we had that huge surge of patients in the UK in March and April, we would have been in really trouble.
Well, it's the unnecessary deaths, because what happens, what people forget. So say it's the unnecessary deaths. So if when you're dealing with the NHS, you're dealing with limited resources. Okay, so you only have limited ITU beds. So if you just let everyone live as they do, as they were, people would just die. Not because they couldn't. They literally, people would die because there were no beds for them to be treated, and there were not enough qualified people to care for them. It would have been horrendous. So actually, as much as I don't like lockdown, and I don't even like the term, because even lockdown is something you do for people in prison. I don't like the term, but it expresses what we needed to do. It would have been horrendous. And the same now. If we don't do anything, we're already seeing, since we've come out of that 4-week lockdown, cases are already going to start to go up. Winter is horrendous as it is in hospitals. So that we worked in hospitals for nearly a decade, and the intensive care units are just filled with people with respiratory viruses throughout the winter, not including COVID. That the beds it's usually full. Anyone who works in ITU, or HDU or anything like that, already. You dread winter. You dread December to March. You absolutely dread it. So adding COVID to that, to let it run free, would be I don't want to say the word that I think, but it would be absolutely absurd. It would be a horrendous thing to do. And there's absolutely no sense. There's no rationale. There's no good reason. The reason is there's no good reason to do that, and it sucks. But you would get tens of thousands of unnecessary deaths because we didn't do anything. And not only that, see, people just count the deaths. You have to think about disability. So if you look into the research about the people now with long-term renal failure, renal disease, people with cardiovascular disease, people with lung disease, these are people who are going to be potentially around the world. We look at millions of people with disabilities now who are not going to be able to function as they used to function.
Absolutely true.
So the deaths are one thing, but it's also long-term short-term and long-term disability as well. And also we don't know a lot of the other long-term outcomes of people who have, because there's people who still experience long-term symptoms for months, but ongoing, like they've had it and they're still experiencing it now. So there's such a spectrum of experiences, but fortunately most people will just get a temperature you know, and a headache, and they're okay. You know, they feel better a few days later, which is great. I'm happy about that. But ultimately, the thing that's going to get us back to normal or near normal living is going to be an effective vaccine, and we're absolutely fortunate that we've got, you know, several good candidates for that, and we're at the forefront of that.
I think there are around 200 in development, but many of them are still in early stages. So,
yeah. So it might even be one that we find out it's going to be even better, even cheaper. But you know, we are where we are, and it's looking pretty promising. So unless there are any more questions come in the next minute or two, maybe we'll start thinking about sort of rounding up. Do you have any book recommendations that people could read up on about vaccines?
I don't actually have any book recommended because I read many papers. Give me 1 second.
That's okay.
Do you have one?
Let me. I actually bought one a couple of days ago, and I haven't got around to reading it yet. And I'm just going to get it up.
I recommend the movie "And the Band Played On," which is a TV movie from 20 years ago, which is on the development of looking at HIV as and the cause of HIV, which is a good insight into how the medical science world works. That was if you find that.
There's a book by Paul Offit called Deadly Choices. That's the book I bought recently. That's meant to be very, very good. Okay, so we've got a question here. Here we go. You don't mind, do you?
No, please. No.
Yeah, so thank you, Richard, for the questions. They're really good questions, really helpful. Okay, so regular flu mutates each year, and just under 500,000 died of it worldwide in 2017-18. Yet no lockdown or anything said on the news. Yeah,
that's true. Yes, I mean, and we had the pandemic strains in 1918, and we had risks. We did actually have a lot. We did look at restrictions though, because when swine flu happened, we did actually do quarantines because there was a major concern about swine flu becoming pandemic. Influenza can be very serious now, and there are a lot of questions about what we can do to prevent influenza pandemic strains occurring. Because some of that often happens because of the combination of influenza viruses between birds, pigs, and humans, and so that brings the question of how do we avoid proximity of those three animals to each other in farming. But no, influenza is very serious. So I think by comparing to influenza and looking at, we can maybe underestimate the severity of COVID because we underestimate the severity of influenza for starters.
Yeah, I guess this is what we're and flu tends to be seasonal as well. The thing about COVID is it's the same all year. It seems like it's the same all year round. You know, the thing about flu as well, it tends to in terms of the mortality rate, it is I guess. Well, actually, I mean, it's broadly kind of reflective in the same that people who are younger tend to not be affected too badly, whereas those who are older are. But I don't think it would. It's
not quite as skewed as COVID, but not quite as.
I mean, we're already at what will be at 1.5 million or something, I think. Over the since March, so we'll probably be close. It's going to be probably over 2 million. So I probably 2.5 million by the time we get to a year, looking like, I reckon. How can this new vaccine be trusted when things like HIV and polio were actually man-made? Well,
HIV and neither of those were man-made. Sorry. There was. I know the HIV. There were questions whether HIV began in a lab or HIV began through in Africa from cells that were infected with HIV given to humans, but neither of those were true. HIV, they're actually, for the sake of information, there are actually two different viruses called HIV. HIV-1 and HIV-2, they have different origins. HIV-1, which is the one we're most familiar with because it affects more of the world, originally came from chimps, and HIV-2 came from macaques.
Yeah. So there's basically, it comes down to evidence. There's no evidence that HIV or polio were man-made. against not from trustworthy sources. Again, you will always find people who will make certain claims. That's just the nature of humanity.
There's no motivation to create a virus like HIV because it will affect everybody.
Yeah. And so it says, why do we already get we get a vaccination for regular flu, yet people still get it? Yeah. So have you got that, Jocelyn? So there's already a vaccination for regular flu, yet people can still get it.
True. But so influenza virus changes relatively quickly. And so when people are, and not the number of strains circulating any one time of influenza, so the vaccines developed against those strains of influenza that are most likely to cause disease and serious disease in people. It is preventative, but it might not have caught all the strains. And it's possible to get a strain that wasn't included in the vaccine. But even then, it's thought that it'll lessen the symptoms. So even if you get any of the strains, it will make it less severe. So that's the answer. It comes down to the continued mutation of influenza virus.
Yeah, and even then, I mean, vaccines are not 100% effective like we want them to be. But you can have a vaccine, but it doesn't mean it's necessarily going to be effective. And that's when we talk about the how effective some of the vaccines have been, the variance between, say, the AstraZeneca one and the Pfizer one.
And part of it comes down to how your body. So when your body sees an infection, it presents different parts of it to the immune system. Says, "Oh, this is something we should attack." And everybody has different responses, so people can display different parts of the virus. And some parts are less good at creating a vaccine response than other parts, or an immune response than other parts.
Yeah. Okay, I think we're approaching the 2-hour mark, so we'll probably have to round up. There's only so many times I can ask questions, but I really appreciate all the questions that have been asked. And if you are on Facebook and you want to carry on the conversation, feel free to get in touch, or the best thing to do will probably be to post comments on YouTube afterwards, and I'm sure Jocelyn and myself will be happy to try and do our best to answer those questions. Obviously we can't stay online, it's nearly 10 o'clock now. But Jocelyn, thank you so much for taking the time to do that. I really appreciate it. And I've learned a lot, as I'm sure those people listening have. And maybe in the new year, maybe we'll get you back for another hour or something if there's no shortage of questions. And I'll try not to do it on such short notice next time, so we can get a lot more questions in advance and things like that. But my hope is that people will watch us afterwards and will find this helpful. But thank you everyone for listening. Thank you for the questions. Thanks again to Jocelyn, and please subscribe if you've enjoyed this. Please comment below, and as I said, we'll try and respond to those questions when we can. And wishing you happy Christmas. I think we've got one more podcast coming out next week before Christmas. And then we haven't got any coming, I think, till the end of January. So me and Phil are gonna have a bit of rest. And as I said, Phil's not here because he's otherwise engaged, having a bit of a rest. So that's why it's just been me this evening. But thank you very much. Take care, and thank you. All right.
Thank you.